Cell staining is technique that can be used to better visualize cells and cell components under a microscope. By using different stains, you can stain certain cell components such as nucleus, cell wall, or even you can stain the whole cell. So most of the stain, they can stain non-living cells, some can stain the living cells, and some of the stains can stain the both live and dead cells. So the question arises, why staining is important? Why we stain the cells? The most basic reason to stain the cell is to enhance their visualization. And this visualization may be of whole cell or the cell components like mitochondria, some particular other section like the nucleus, or some other cell organelles. So this staining can be done to highlight the metabolic process or to differentiate between live or dead cell. In later part of this discussion, I will let you know how to stain the live and dead cell. So before going to the staining protocol, I wanted to share the names of different stain that we commonly use in cell culture lab. So here are some common stains that we use in our lab. Here you can see the first one is Bismarck brown. This dye, color acid mucin, a type of protein, gives the yellow color and may be used to stain the live cells. The other dye is carmine, colors glycogen or animal starch and give the red color. Comessi blue. This is a brilliant blue color stain, as its name is showing already, and it is often used in gel electrophoresis. Crystal violet stain. This stain the cell wall purple, and this stain is used in gram staining. DAPI, a fluorescent nuclear stain that stain the DNA in the living cells. Eosin, a counter stain to hematoxylin. This stain colors red blood cells, cytoplasmic material, cell membranes, and extracellular structure, and the color may be pink or red. Ethidium bromide. This stain colors unhealthy cells in the final stage of apoptosis, or the cells which are near to cell death, and this gives fluorescent red to orange color. Fuchsin stain, as you can see here at the second last line of this slide. Fuchsin. This stain is used to stain the collagen, smooth muscle, or mitochondria. Hematoxylin, a nuclear stain. This stain the nuclei, giving the blue to violet or brown color. Here is the list of some other common stains. On the top you can see Hox staining. The Hox stain, it stains the DNA in living cell. Iodine stain, used as a starch indicator. When in solution, starch and iodine turn a dark blue color. The third one here in this slide is malachite green stain. This is blue-green counter stain. And this can be used to stain the bacteria or spores. Methylene blue. This stains animal cell to make the nuclei more visible. Toluene red stains the nuclei red and can be used for the living cell staining, nuclei staining. Neil blue stain the nuclei blue. Neil red or Neil blue oxazone this is used to stain the lipid, as you can see here, and it gives the red color. Osmium tetroxide, also used to stain the lipid. Rhodamine stain. This is protein specific fluorescent stain used in fluorescence microscopy. Safranin, a nuclear stain used as a counter stain to color the collagen, and it gives the yellow color. When you are done with the staining process, you can store the stain slides at low temperature in refrigerator, and you can also observe them microscope whenever required. Here is some important terms used for the cell staining. The cell staining techniques and preparation depend on the type of stain and analysis used. We can follow the following one or more than one of the given procedures. Here you can see the permeabilization treatment of cells, generally with a mild surfactant which dissolves the cell membrane in order to allow larger dye molecule to enter inside the cell. So when we are using the dye having the big structure, to pass through the cell membrane they need, of course, a wide path from the cell membrane. So this process actually help that particular dye to pass through the cell membrane. Fixation. Fixation serves to fix or preserve cell or tissue morphology during the preparation process. This process may involve several steps, but most fixation procedures involve adding a chemical fixative that create chemical bonds between proteins to increase their rigidity. Why we need that? Because the dyeing process, the staining process, is multi-step process, and during these process the cell can damage. To reduce this damage, we use this fixation process. We use these fixative chemicals, which are formaldehyde, ethanol, methanol, or picric acid. Mounting, slide mounting. This involves attaching the sample to a glass, that is of course the substrate of your cell culture. For this, cell may be grown directly to the slide, or loose cells can be applied to a slide using a sterile techniques. So in this process, very thin slices of the material, I mean your sample, such as tissue, may also be applied to a microscope slide for observation. Staining. Application of the stain to a sample to color the cells, tissues, components like mitochondria, nuclei, or metabolic processes. This process may involve immersing the sample in a dye solution and then rinsing and observing the sample under a microscope. So the handling of microscope is very important when you are working with cell staining. You need to understand the handling process required for fluorescent microscope, because you use fluorescent microscope frequently when you are dealing with cell staining. So in this slide I will give you an overview how we use these stains to stain or dye the cells. You have seen many cell staining reagents, and one of them is FDA, fluorescent diacetate. We call it FDA, and this is green in color and it stains alive cells. And the PI is propydium iodide. This gives red color to the dead cell. This PI stain the nuclei of dead cell. So FDA and PI can be applied to check the live and dead cell in your sample. For example, you have the cell culture and you want to test any drug's killing effect. For this, you will seed the cells and then after appropriate incubation you will treat with that particular drug. Again you will incubate for the desired time. Let's suppose for 12 hours. And after 12-hour incubation, you can stain your cell sample with FDA and PEI stain to check the alive and dead cells. Previously, you discussed the MTT assay and hemocytometer method to check the alive cells, and of course you can find the dead cell also using the MTT and hemocytometer. How? Just you need to minus the live cell from the total number of cells from control, then you can get the dead cell. But the FDA and PI is different method, a staining method to check the live and dead cell in your sample. So in this way you can have the visual evidence of your sample. So this assay is fluorescent space live dead assay and can be used to evaluate the viability of mammalian cells. Simultaneously, use of two fluorescent dyes allows a two-color discrimination of the population of living cells from the dead cell population. In this assay, we will discuss a staining protocol using FDA and PI, which stain the viable cells and dead cells respectively. This staining protocol is applicable to adherent cell attached cells like the fibroblast cells, keratinocyte cells, you already know, the melanoma cells, single cell embedded in extracellular matrix, and 3D cell cluster. That we will discuss what is 3D cell cluster later on. You already know the embedded cell culture, how to culture, and you are already familiar with this. First we will discuss how we can stain adherent cells with FDA and PI. Before discussing the protocol for staining, we need to understand how this FDA and PI stain work, how they stain the cells in our sample. As I said, the FDA is the stain which gives the green color to the live cells. This FDA is taken up by the cells, live cells, which convert the non-fluorescent FDA into the green fluorescent metabolite fluorescein. This fluorescein we can observe under microscope, and this gives green color to our sample if the cells are alive. The measured signals serve as an indicator for live or viable cells, as a conversion is esterase dependent, which is of course observed in live cells. However, the PI is red color. This is nuclei staining dye. PI cannot pass through a viable cell membrane. So if the cell is alive, this PI cannot penetrate, and if this cannot penetrate in a live cell, it means it cannot give red color to the live cell. It penetrates through the distorted area of the cell membrane, the broken or damaged area of the cell membrane, and intercalate with DNA double helix and gives red color. So the cells which have the damaged cell membrane, they allow this PI to penetrate, and this interact with the DNA, giving the red color. So using this FDA we can check the live cells in a population, and using PI we can see the dead cell in our population. So when we purchase this FDA and PI kit, we get this FDA and PI vials, and we will make their solution like FDA 5 mg per ml and PI 2 mg per ml, and their volume for each sample depend upon the area of the sample. Of course, if we are using the small area, if we need to stain the small area of our sample, then we need less volume, and if we are having the huge area of our sample to stain, then of course we need to use the high volume. However, whenever we want to stain our sample, we need to prepare this FDA and PI fresh, and we should store it at 4 Celsius. And this is light sensitive. We must need to protect it from light, and we should not put it on the room temperature for a long time. And of course, we cannot use it again and again. This is the reason we need to prepare this fresh every time whenever we need to stain. If we are using the old one, it will give the poor signal, and the staining will be blurred. Even our sample have the live cell, FDA maybe cannot give the clear image if we are using too old, or if we are not protecting our sample after staining from light. So here is the staining protocol for adherent cells. Like if you have, for example, fibroblast cells that are grown on the glass slide or on the bottom of well plate, how you will proceed for FDA and PI staining. Here is the recipe. First, you need to prepare the staining solution. This is number 1, and keep in refrigerator. Of course, it is temperature sensitive. You need to save it in a 58 temperature. Then removal of cell culture medium. So here we assume that the sample that you are going to stain is already treated with the stimulus like drug, plasma, or whatever you want to test. And now finally you are at the stage where you want to stain and check that stimulus effect, that cells are alive or dead. So your sample is at that state. Then you will remove the cell culture medium. Then you will add the staining solution. And of course the volume depends upon the geometry of the used slide or your sample. If you are using small glass slide, then the volume you need for the staining is very less. However, if you want to stain the whole culture dish, that is very big in size as compared to the for example, cover slip, in that case you need huge volume. But of course you can use the small cover slip, small well, or even the big culture dish according to your research design. And after addition of the stain, you need to incubate the sample at room temperature for 4 to 5 minutes in dark. The reason to store in dark is the PI and FDA are light sensitive. After incubation, you need to remove the staining solution. You need to wash the staining solution with PBS, and then finally after 2 to 3 time washing, you can put your sample in media without FCS, fetal calf serum. So you can use media like MEM, DMEM, or you can use PBS. We prefer usually the PBS because it is transparent and we can have the good images when we are observing our sample under fluorescent microscope. And when you are done with all these steps, you can see the clear image under microscope. So this is how the staining look like. Here in the bottom you can see the 4 images. 1, 2, 3, 4. Yes, here. So here this one, left to right, this one, the first one, is phase contrast image of your sample. And this one, the second one, is PI signal. So which are just 1 or 2, you can see here. It means very few cells are dead in your sample. This one is FDA signal, which represents the live population. So most of your sample population is alive in this case. So this is just, negligible killing the RPI signal. And this one, FDA and PI signal, both. So you can just, this one you can see here, small red dot. That is showing the cell is dead. However, the rest whole green population is alive. In this picture you can see the green and red clearly. This green population is the population which is alive, and the red color is showing the PI staining, which represents the dead cells. So here you can see few dead cells. So this image is independent of this image. This image has no relation with this image. I just put this image to show you how this red and PI look like, and how to make you clear, I just put this extra image. So this is the live and dead FDA PI staining for adherent cells. Okay, students, let's extend our discussion of cell staining. Today we will discuss further the cell staining and tissue staining, and I will show you some publication where the cell staining and tissue staining data is reported. So you can learn how this staining data can be presented and can be used for the publication. Of course, that is the main goal of one of the goal of your research. So first of all, here I would like to show you this paper. This paper is published by the Common Penguin in the journal Cell Death and Disease in 2013. The title of this research work was Preferential killing of human lung cancer cell lines with mitochondrial dysfunction by non-thermal dielectric barrier discharge plasma. So if we just quickly review their work, what they did is they used lung cancer cell lines and healthy lung fibroblast or other healthy lung cell lines, and they treated healthy and cancer cell line with non-thermal plasma, and they checked the killing effect of plasma. Either it is more efficient in killing for cancer cells or healthy cells. This was their goal to uh, investigate during this research work. So during their research work, they stained the cell to observe the morphology of the cell, and they also stained the mitochondria. Our topic of discussion is the staining, so we will check how they stain the cell for morphology and how they stain the mitochondria. So this is the staining data. We will come to the discussion, the explanation of this data later. First, I wanted to show you how they look like, the picture and the staining data look like. However, first of all, we would like to see how they perform this staining. And let's go on the material and method section and see how they stain this. I hope this highlighted text is visible to you, right? बच्चों आपको ये highlighted text visible है?
Yes, sir.
Okay. So here what they did, they stained the actin fibers to see the cell morphology, and of course they stained the mitochondria as well to see the changes in mitochondria. Before studying this, we need to understand what is the status of their sample at this stage. They culture the cell, lung cancer and lung healthy cells. They let them incubate and attach. After appropriate incubation, they treat it with plasma with different exposure times, 1, 3, 5 minutes, for example. And then again, they incubated the sample, and after appropriate time, like 24 hours, they start the staining process. So their sample is plasma treated sample, and now they wanted to stain and see what is the plasma effect on the cell morphology and mitochondrial shape structure. To investigate this, they use the staining technique. So let's read this. Cellular and mitochondrial morphology. This is basically the staining. Actin fibers and mitochondria were stained after 24-hour plasma treatment. Cells on cover glass were washed with DPBS, fixed with paraformaldehyde for 15 minutes, and permeabilized by Titan X100. After washing with PBS 3 times, actin fibers were stained by Faladin dye. This is the staining they use for the actin fiber staining. After washing with PBS 3 times, mitochondria were stained by the Mitotracker dye solution. Then the same sample they use for the mitochondrial staining. So the sample is same. They use 2 dye to stain the actin fiber and mitochondria. For actin fiber, they use Faladin dye, and for mitochondrial staining, they use Mitotracker dye solution. And when they are done with the staining, they observed the sample under fluorescent microscope. And under fluorescent microscope, how the sample look like, I'm gonna show you the taken pictures. Okay, now this staining data is available, is visible to you on your uh, laptop screens. स्टूडेंट्स, आपको आवाज मेरी पहुंच रही है? ये डाटा आपको
Yes,
विजिबल
sir.
है आपकी स्क्रीन पे?
Yes, sir.
Sir, नजर आ
Yes, sir.
ओके, तो यहाँ पे बेटा देखें, they used for H460, that is the lung cancer cell line, and they use MRC5, that is the healthy lung fibroblast cell line. They use the cancer lung cell line and lung healthy cell line, so it is good comparison. whenever you want to test any drug effect or any stimulus effect, especially for killing or cytotoxicity, so then it is always good to use the same organs, cancer and healthy cell line for better comparison. They did the same here. H460, which is the cancer cell line, and MRC5, which is the healthy lung fibroblast cell line, they used, and they got the better comparison. So here you can see this is the 1 minute no plasma treatment. They represented 0 minute mean. This is the control sample. This sample is treated is not treated with anything like plasma or any stimulus. However, this sample you can see is treated with 1 minute plasma, and this one 3 minute, this one 5 minute. The case is same for this cell line as well. So this A and C images are actually the stain data, and B and D is actually the magnified portion of this data. Like this B, this image is the magnified section from here. The case is same for here, here, and other pictures as well. So let's discuss this sample, this image. The red color is actin, which is stained with Faladin. And in morphology, you can see here in the treated sample, and you can compare with this control sample. So this sample, you can see this is the cell. And if you look at this cell, the boundary of the cell membrane, which is bumpy, which is, not regular, uneven cell membrane you can see here as compared to here in control. So this is the morphological changes after plasma exposure. It means by using the staining technique, you can observe the change in morphology as you did here. And the case is same for this MRC5 cells. You can see this is the control sample. And this one is 5 minute treated sample. And this treated sample, the cells are more wider than this control one. In control, the cells are mostly long, right? However, here they are more or less spherical. They are broader in shape. So you can check the morphology by using the actin staining, as they did in this data. And this green staining is the staining for mitochondria. And you can see this is one cell, this one is second, this is third cell, fourth cell, fifth cell. So this green signals are coming from mitochondria. And this, green signal you can see here, and in this sample you can see here, and they are tubular in shape. But if you look down at the 5 minute plasma treated sample in this image, the same mitochondria in the sample, they are not in tubular form. they are swollen as compared to the control, and they become small fragments, as you can see here. But in control, they are not in, like this. They are not swollen as in here in this sample. And in this sample, 5-minute plasma treated sample, the mitochondria are not the way they were in control, like they were in tubular form. You can see they were arranged around the nucleus. Here in the middle is nucleus, and these mitochondria were arranged around the nucleus. However, in treated sample, they are scattered. You can see here. So we cannot really observe the tubular appearance of the mitochondria around the nucleus in this sample. So from this, you understood that by applying this staining technique, we can see the morphological changes in mitochondria. And these images are for H460, and here we can see in case of healthy uh, cell line, that is MRC5. If you look at this sample, the control sample, here also the mitochondria are arranged around the nucleus. Here in the middle is nucleus. Here is the magnified form. This circle here is the arrangement of the mitochondria, and they look in tubular form, right? I mean, arranged around the nucleus. Here in the middle is nucleus. And in 1 minute, same, we have to some extent similar arrangement of the mitochondria. Here in 3-minute sample is also, but here you can see the mitochondria are very small in size. They are not swollen like this sample. They are, in fragments, very small fragments. They are not swollen at all. And they look quite different from this controlled sample. So from this data, you can conclude that using the staining technique, we can track the mitochondrial morphological changes. So this is the staining data, and you need to understand how we arrange this, how we present this, how we add this in the publication like this one. Now I will share you another publication where they stain the tissue with some dyes, and you can learn how we can collect the staining data, how we gather or extract information from that particular data, and how we express it. So here, this is the paper where the researchers did work on the plasma activated solution and check the melanin production. The plasma here is the non-thermal plasma, which produce reactive oxygen, reactive nitrogen species, UV radiations, ozone, and many more, the small electrons, molecules, atoms. And especially these reactive species are related with, can influence the synthesis of melanin. So in this paper, after plasma treatment, the melanin content in cell B16F10 cell, the content was monitored, and later on they use the same plasma treated solution on the tail of mouse, took the biopsy sample and stained that tissue with melanin staining dye and observe the melanin content in the skin. So during their work, they found that after plasma treatment, the melanin content is increased in the cell. They wanted to see if this is possible in mouse model as well. For this, they treated the mouse tail with plasma activated solution and they stained it with Fontana Methane staining, which is specialized for melanin staining. So how they proceed for this staining, let's see at the material and method section. Now, Fontana Methane staining. To analyze the melanin content in the mouse tail skin, tail biopsies were obtained after completing the final treatment of plasma activated solution. Plasma activated solution mean they treated water or buffer with plasma and they use that plasma treated buffer as a plasma treatment and call it plasma activated solution and use for their experiment. Now, after treatment, they wanted to see the melanin content. That's why they are performing this staining. Now, to analyze the melanin content in the mouse tail skin, tail biopsies were obtained after completing the final treatment of PAS, plasma activated solution. After the fixation in formalin for 24 hours, samples were embedded in paraffin. For Fontana Mason staining of the skin section, paraffin tail section were deparaffinized and hydrated in distilled water. Fontana Mason staining was performed as described in the manual of this kit. Briefly, the method they use is explained over here. The slides were immersed in freshly prepared ammoniacal silver solution, and this solution come with the kit. The reaction was stopped when the tissue section become yellowish brown with colorless background. Then the samples were rinsed in several changes of distilled water and then immersed in gold chloride solution. This also comes in the Fontana Mason staining kit. The sections were rinsed in several changes of distilled water and then placed in sodium thiosulfate solution. The sections were rinsed again in distilled water and then counter-stained in nuclear fast red for 5 minutes. The slides were washed with water, and then dehydrated with 3 changes of fresh absolute alcohol. Finally, the slides were mounted and the pictures were taken under fluorescent microscope. How that picture looked like, I'm gonna show you here. Okay, this is that stain tail section of uh, control mouse and plasma treated mouse. This UN mean control sample, which is not treated with plasma. And here PAS 9 mean plasma activated solution treated with 9-minute plasma. And this PAS 15-minute plasma treated solution. Here in control, you can see the arrowhead. These arrowhead, indicate the brown or black color present in the outermost layer. Here, this is the melanin. You can see here as well. This is very less as compared to the treated sample, PAS 9. Here, for example, you can see the more dark layer of the melanin. So here after PAS, there is more melanin content as compared to the control. It means plasma is increasing the melanin content. And this also means that Fontana Mason staining is staining perfectly the melanin content. It is very clear from these images. And here too you can see some dark layer, which is of course melanin. So here what you learn is you can stain particular proteins like melanin as well in tissue sample. And this staining technique is very helpful for collecting the useful data. As they wanted to study the plasma effect for melanin, and this staining technique helped and give very useful data. As you can see here, this is the visual data which have very good impact on the reader, and it is very easy to understand, and it is very reader friendly as well, because we can see the actual data with our own eyes. As you can see here in control, very less melanin, here more melanin. So this solution, this staining technique is very helpful. Here I will show you another paper which is also showing the human skin staining. In this publication, the author used metformin, that is, anti-diabetic drug, for the melanin inhibition to investigate the modulation in melanogenesis. This metformin is widely used for type 2 diabetes, and it is also known for the inhibition of cancer proliferation, and also cyclic AMP. And this cyclic AMP is um, closely associated with melanin modulation. So they use this metformin to investigate either it can uh, change the melanin content or not. So here are some images. Okay, we will discuss this area only for easy understanding. This sample is a human skin sample and control, and this one is the human skin biopsy sample treated with metformin. As you can see here, this is the relatively lighter in color. The color tone of this sample is light, and this is comparatively dark. And they use the same Fontana Mason staining technique as we discussed in the previous paper, and they got these images. This is for control sample. Here you can see the black uh, uh, pigment, which is the melanin, and after metformin treatment, as you can see here, is relatively less melanin content, and we can see here we have less uh, melanin content after metformin treatment. So here we also got the visual data using the staining technique, which is very helpful for understanding for the reader. So do you have any question about cell staining or tissue staining? स्टूडेंट्स आपको आवाज आ रही?
Yes, sir.
जी, आपको कुछ पूछना है यहाँ तक?
No, sir.
Okay.